Hello, ladies and gentlemen. Today we're gonna talk about signal transduction. In this chapter, we're gonna talk about G proteins, how they activate an enzyme called adenylate cyclase. We're gonna talk also about the receptors coupled to these G proteins, the structure and function, and the accessory proteins required for the function of G proteins. Next, we're gonna talk about a certain toxin and how they affect the G proteins and how to stop the responsiveness of a signaling system by something called desensitization. The next part is gonna be about the tyrosine kinase based signaling, and the last part is gonna be about phosphoinositide cascade. Let's start by explaining the meaning of signal transduction. Well, signal transduction, also known as cell signaling, is the transmission of an extracellular signal to the cytosol of a cell leading to a certain response. So how does it occur? First of all, the agonist, also known as the ligand, is received by a cell when it binds to a cell surface receptor such as the G protein coupled receptor. Then, it triggers a signaling cascade inside the cell to finally lead to a cellular response. G proteins are heterotrimeric, which means they are made of 3 subunits, that's why it's called trimeric, and hetero because the 3 subunits are not the same, they are not identical, they are different one from the other. So the G proteins are made of 3 subunits, the alpha, beta, and gamma subunits. The alpha subunit is attached to either a GTP or GDP. When GDP is attached to G protein, the G protein is inactive. Upon the binding of a ligand to the G protein coupled receptor, G protein become activated, so GDP is replaced by GTP. Let me explain to you how. When there is no ligand bound to a receptor, G protein is in its inactive state with a GDP bound to its alpha subunit. Upon the binding of a ligand, alpha subunit exchange GDP for GTP. So now alpha has a GTP attached to it, which activate G protein. Upon the activation of G protein, an enzyme found on the membrane is activated. This enzyme, depending on its nature, is gonna have some activity. I'm not gonna talk about the activity of this enzyme mentioned here because I'm gonna talk about it in the next slide. But what I need you to know that when the action of the ligand must be terminated, G protein with its GTPase activity is gonna hydrolyze the GTP into GDP and inorganic phosphate. So now now alpha has a GDP bound to it, which inactivate again the G protein. During exercise or when you're fasting, the glycogen in your body must be broken into glucose. So 2 hormones come to your mind, the glucagon and the epinephrine. These hormones control the glycogen metabolism by binding to a G protein coupled receptor and activating the adenylate cyclase enzyme. So what happens is when the hormone binds to the receptor coupled to G protein, alpha exchange its GDP with a GTP and become activated and activate the adenylate cyclase enzyme. This enzyme hydrolyzes ATP into cyclic AMP. Cyclic AMP is known as the second messenger. It triggers a signaling cascade that amplifies the initial signal so that one hormone can lead to the activation of many cyclic AMP, and this is known as the signal transduction. So the cyclic AMP is gonna activate protein kinase A, which is gonna activate or gonna phosphorylate different proteins, leading to control the activity of glycogen phosphorylase and glycogen synthase to promote the degradation of glycogen into glucose. Of course, I'm not asking you to memorize these enzymes because this is not the topic of our chapter. What I need you to understand that the G protein, once it's activated, having a GTP bound to its alpha subunit, is gonna activate the adenylate cyclase that's gonna hydrolyze ATP into a second messenger, the cyclic AMP, and a lot of molecules of cyclic AMP will be produced, and these molecules gonna activate protein kinase A, which gonna control the activity of glycogen phosphorylase and the glycogen synthase to promote the degradation of glycogen into glucose. Now, what are the G protein coupled receptors that I've been mentioning earlier? The G protein coupled receptors, also called GPCRs, are heptahelical receptors, which means they are made of 7 alpha helices. They are also called 7TM or 7 transmembrane receptors because they pass through the cell membrane 7 times. They have an extracellular side and a cytoplasmic side. To this receptor, we have a G protein coupled to it. That's why it's called a G protein coupled receptor. So like the figure below, we have the 7 transmembrane receptor, and coupled to it we have a G protein made of the 3 subunit, the heterotrimeric G protein made of alpha, beta, and gamma subunit, and in its inactive state we have a GDP bound to the alpha subunit. So the G protein coupled receptor is the type of receptor that's gonna bind a certain ligand to lead to a certain cellular response. What kind of agonists or ligands bind to these GPCRs? Well, the nucleoside, the nucleotides, the calcium, the catecholamines like epinephrine, norepinephrine, dopamine, and other biogenic amines like histamine, serotonin, and the eicosanoids, mediators of inflammation like the prostaglandin, prostacyclin, thromboxane, leukotrienes, and lipoxins, which are derivatives of fatty acid, the arachidonic acid, bind to this type of receptors known as the GPCRs. They also constitute the receptors of olfactory and gustatory molecules, as well as to several light-sensing proteins in the retina like the rhodopsins. Also, they constitute important class of drug targets because 50% of the drugs approved exert their therapeutic effects by selectively interacting with specific G protein coupled receptors. So far, we know that G protein is heterotrimeric, made of 3 subunits, alpha, beta, and gamma. Alpha has GDP in its inactive state, and when the ligand binds to the G protein coupled receptor, GDP is exchanged for GTP. So we have these 3 subunits, alpha, beta, and gamma, bound to each other, and they are anchored or attached to the inner surface of the membrane. G beta and G gamma bind one another with high affinity. That's why we refer to them as a complex G beta gamma. G alpha is covalently attached to the membrane through myristylation or palmitylation, the kind of lipid modification by attaching a myristic acid or palmitic acid, or maybe both. The G beta gamma subunits, they are attached to the inner surface of the membrane through prenylation of the G gamma. These lipid modifications stabilize the interaction of G beta gamma with G alpha and together with the membrane. So what happens when the ligand binds to the G protein coupled receptor? G protein becomes activated, GDP is exchanged for GTP, there is a dissociation of the G alpha from beta gamma, G alpha with GDP activate an effector protein, which is in this case the adenylate cyclase. Not only G-alpha activate downstream effector proteins, G-beta gamma can also activate a wide variety of signaling proteins. It can activate several isoforms of adenylate cyclase. It can also activate certain ion channels, sodium, potassium, and calcium ion channels. It can also activate protein tyrosine kinase and phospholipase C-beta component of phosphoinositide signaling system that we're going to talk about in the last part of this chapter. G proteins are not only able to activate adenylate cyclase, like the case of glucagon or epinephrine binding to the G protein coupled receptor and activating adenylate cyclase. That G protein is a G stimulatory because it activates the adenylate cyclase. But we have also another form of G protein that can lead to an inhibition of adenylate cyclase. This G protein is known as an inhibitory G protein or GI. GI may have the same beta and gamma subunit as does GS or the stimulatory protein, but it has a different alpha subunit. So the GI alpha inhibit adenylate cyclase, whereas GS alpha activate adenylate cyclase. An example of this are the receptors for somatostatin and opioids that are coupled to a G inhibitory protein. So to summarize this part, if we have a stimulatory external signal that binds to a receptor coupled G protein, G alpha S stimulatory, the alpha subunit exchange GDP for GTP, it dissociates from the beta gamma subunit and activate adenylate cyclase. The adenylate cyclase activated is going to hydrolyze ATP into cyclic AMP and the signal is amplified. Then the cyclic AMP is going to activate a protein kinase A, which is going to phosphorylate other proteins, leading to a cellular response. This is in the case of a stimulatory external signal. If the signal must be terminated, or to terminate the action of this external signal, the G protein with its GTPase activity hydrolyzed GTP into GDP and phosphate. G alpha GDP dissociate from the adenylate cyclase and reassociate with G beta gamma to reform the inactive G protein. Now, this is for the part of the G alpha or the this is the part of the GS or the G stimulatory G protein. Now, for the G inhibitory or the inhibitory G protein, it responds to an inhibitory external signal that when it binds to the receptor coupled G protein, GI, the alpha subunit exchange GDP for GTP. They dissociate from beta and gamma. The G alpha I GTP inhibit now the adenylate cyclase. In addition to GI and GS, we have different members of G protein family that have different effectors other than the adenylate cyclase. Let's mention some of them. GQ alpha is a member of G protein that forms a link in the phosphoinositide signaling system with the phospholipase C as effector protein. Of course, I'm not gonna go into details at this part of the chapter, but at the end of it, we're gonna talk in details about the phosphoinositide signaling system. The second member of G protein family is transduction, known as GT alpha. It's a variant of GI alpha, which transduce visual stimuli by coupling the light-induced conformational change of rhodopsin to the activation of a specific phosphodiesterase (PDE), which then hydrolyzes cyclic GMP to GMP. In fact, what happens here is that the light leads to a conformational change in rhodopsin, which in turn lead to the activation of transducin, which is a GT alpha. Transducin dissociate the GT alpha, exchange GDP for GTP. The alpha GTP dissociate from beta gamma and stop the inhibition of the phosphodiesterase, thus activating the phosphodiesterase, which result in the breakdown of cyclic GMP to GMP. As a result, the cyclic GMP-gated sodium and calcium channels close, thereby triggering a nerve impulse indicating that light has been detected. T-alpha is a variant of stimulatory G protein, which is expressed only in olfactory sensory neurons and participate in odorant signal transduction. And finally, we have G12 alpha and G13 alpha that participate in the regulation of the cytoskeleton. In order to function properly, G proteins often require accessory proteins like GAP, the GTPase activating protein. This protein activates the hydrolysis of GTP. Cyclic GMP phosphodiesterase and phospholipase C beta exhibit GAP or GAP activities toward GT alpha and GQ alpha, so it catalyzes the hydrolysis of GTP into GDP. The second accessory protein is GEF, the guanine nucleotide exchange factor. It promotes the exchange of GDP for GTP, as you can see in the figure. The third factor is GDI, the guanine nucleotide dissociation inhibitor. So it binds to GDP form and prevents its exchange into GTP. Cholera is a life-threatening disease marked by severe diarrhea. It is caused by a bacteria called Vibrio cholerae. In the intestinal cells, the bacteria secrete cholera toxin, which is made of two types of subunits, A and B. There is 5 copies of B and 1 copy of A. B helps in binding to the host cell receptors and A have the toxic activity. Cholera is a AB5 toxin, and because it specifically affects the intestine, it's called enterotoxin. Now each of the 5B subunits bind to a ganglioside GM1 on the intestinal cell membrane. The A subunit consists of 2 fragments, A1 which contain the toxin activity and A2 which assembles A and B subunits. Before the cholera toxin secretion, A subunit is cleaved by a protease into 2 fragments, A1 and A2, joined by SS bonds. Now the binding of cholera toxin to ganglioside GM1 triggers endocytosis. The vesicle is transported to the ER. Then the A1 peptide is released from A2 by a reduction of SS bonds. From the ER, the toxin A1 is then transported into the cytoplasm. The mission of A1 is to modify the G protein that controls the activity of adenylate cyclase. Now, A1 uses a molecule of NAD+ made of nicotinamide and ribose sugar and ADP. So it catalyzes an irreversible transfer of ADP ribose to the arginine side chain of G-alpha subunit. This is an ADP ribosylation reaction catalyzed by ADP ribosylation factor ARF in complex with GTP. This reaction prevents the G protein from cleaving GTP into GDP and inorganic phosphate. So the adenylate cyclase remains activated for a long period, increasing the level of cyclic AMP. The high level of cyclic AMP or the cyclic AMP is gonna stimulate a protein kinase A that stimulate a chloride channel to open. Once the chloride channel open, the chlorides and other ions leave the cell. Water also leaves the cell in order to equilibrate or in attempt to equilibrate the osmolarity. The bloodstream provides the water and chloride and other ions that enters the intestinal cell, the intestinal lumen. The influx of fluids leads to diarrhea. Another toxin that affects the G protein is the pertussis toxin. This toxin is secreted by the bacteria Bordetella pertussis that causes the whooping cough. This bacteria secretes an AB5 toxin that ADP ribosylates a specific cytosine residue in GI alpha, the G inhibitory alpha. So what's affected here is not G alpha as like in the case of cholera toxin, but it's the GI alpha that normally inhibit the adenylate cyclase. This toxin ADP ribosylate a cysteine residue on G-alpha I and prevent it from exchanging its bound GDP with GTP, and therefore it prevents the GI alpha from becoming active to inhibit the adenylate cyclase. Thus, it cannot inhibit adenylate cyclase, and thus adenylate cyclase continue to hydrolyze ATP into cyclic AMP. The high level of cyclic AMP weakens the cellular defense and increases the susceptibility for infection. A possible treatment for this type of toxin is the phosphodiesterase, because the phosphodiesterase is an enzyme that hydrolyzes the cyclic AMP into AMP. Thus, it lowers the cyclic AMP levels in the body, and this will solve the problem.